Alrighty. So at this point, whenever you're ready, Pepe.
Welcome, everyone. I'm Pepe Ping from the Training, Recruitment, and Mentoring Working Group of the Bridge2AI program, Bridge Center at UCLA.
Welcome, everyone. I'm Jack Rincon, project manager for the Training, Recruitment, and Mentoring Working Group at Bridge2AI. My role involves coordinating AI-focused biomedical training, ensuring a strong foundation in both technology and ethics for current and future scientists. I also assist in managing Dr. Pepe Ping's lab, assisting with various initiatives from publications to outreach for Bridge2AI. Dr. Pepe Ping, a professor of physiology, medicine, cardiology, and bioinformatics at UCLA. It's been a pleasure to work with her. We're committed to supporting the TRM efforts at Bridge2AI, and Dr. Ping will now take the lead to officially open today's session.
Thank you, Jack. Welcome, all of you. We're launching the first in a series of lectures from our Bridge2AI training resource, which we call the Grand Challenges Education Module. This series will provide insights into datasets across our entire consortium. The very first lecture goes to the introduction of Salutogenesis datasets. This is a flagship project in the context of our Grand Challenge projects. The TRM working group's goal with this particular module and the series of 4 lectures is to prepare our Bridge2AI scholars, our investigators, as well as the public to engage with Bridge2AI consortium AI-READI biomedical datasets, understanding their data structure and their potential applications in the broad biomedical field as well as clinical care.
All right. The focus of today's lecture is on AI-READI project and its role in studying salutogenesis in type 2 diabetes mellitus, T2DM. We are honored to have 3 esteemed presenters, Dr. Cecilia Lee, Program Director, Dr. Sally Baxter, Principal Investigator, and Dr. Bhavesh Patel, who's Co-Principal Investigator. Together, they will provide an overview of AI-READI datasets, a critical component of Bridge2AI. Dr. Cecilia Lee, a clinician scientist from the University of Washington, specializes in ophthalmology and AI applications in healthcare. As program director for AI-READI, Dr. Lee oversees a project aimed at creating an ethically sourced AI-READI dataset for type 2 diabetes. Her team is collecting data from over 4,000 participants to build a multimodal atlas that will support AI-driven research, offering insights into health transitions in diabetes patients.
Also presenting today is Professor Sally Baxter. She is the Chief of Ophthalmology Informatics and Data Science at UC San Diego. She has pioneered and leveraged AI to improve clinical outcomes and health equity in the ophthalmology care, particularly in underserved communities. Dr. Baxter's work, broadly supported by NIH awards, emphasize the development of AI-READI datasets that reflect diverse populations, ensuring that AI advancements in healthcare are inclusive and equitable.
Dr. Bhavesh Patel is a research professor at CalME 2 for specializing in computational modeling and fair data practices. He leads the Fair Data Innovations Hub, where he promotes data sharing and accessibility standards that empower biomedical researchers. His contributions to the AI-READI project focus on establishing fair compliant data sets that enhance research reproducibility and collaboration.
We're hoping by the closure of this lecture, you will gain an understanding of the mission and scientific goals of the Salutogenesis Grand Challenge. learn about the AI-ready datasets creation and ethical considerations, as well as explore opportunities to collaborate with this grand challenge within Bridge2AI community and beyond. With that, I will hand it over to our distinguished speakers. Dr. Lee, please.
Thank you. Thank you so much, Dr. Ping and then Jack. It has been really, really an honor and then a pleasure to work with you. And then so we're going to get started. So I'm very excited to kind of start this lecture off about introducing our data generation project on Salutogenesis. And please do let me know if you have any question at the end. Okay, so I wanted to, since this is the first time we are presenting in terms of the Bridge2AI consortium, I wanted to briefly mention about the goal and the main purpose of the Bridge2AI consortium. In terms of we have had, for past several years, we had tremendous progress in machine learning and artificial intelligence, and then really realizing the power of AI in the research of healthcare and then biomedical fields has been really impressive. But NIH also recognized that there is really an urgent need to create a data that is AI ready, that can really propel the study, at the same time integrating ethics and then people to be able to make these efforts happen. So Bridge2AI consortium really is divided into more of a data, ethics, and people framework to understand that these efforts really need to be combined, integrated efforts. And if you are thinking of the scientific discovery pipeline, can you see my my arrow, my pointer here? Okay, great. And then we're like right here. The Bridge2AI is about preparing the data, collecting the data, so that it can be used for future model development and future model evaluation. And then this is really essentially the scientific discovery pipeline. So there are two major components of Bridge2AI consortium. One is Bridge Center, where they focus on integrating all the work, disseminating all the results and outreach, and then evaluating, and then really being able to come up with best practices, and then also really engaging with outside stakeholders in community. So this TRM work group kind of really is a representative of like all the efforts that they are doing. And then we are very grateful to be invited to provide this lecture. So there are 4 data generation projects in the Bridge2AI project in the consortium. And then ours is here, Salutogenesis, AI-ready restoring health after disease. And we'll kind of go over more in details, but I wanted to show that there is various different projects among the Bridge2AI program. So what is our mission of our project? It's to create a multidimensional ethically sourced data set in diverse people for studying salutogenesis in type 2 diabetes. And we call ourselves AI-READI project. What is salutogenesis? So in medical field, in biomedical field, we are very familiar with the concept of pathogenesis of one going from healthy state to disease stage. And then we always think about risk factors. What are the environmental, genetic, and behavioral risk factors? However, the reverse is really the salutogenesis. What are the factors that we need to work on to promote health, to push people from disease state to health? So that is what we are trying to study in type 2 diabetes using our data set. But obviously, that will in turn allow us to study the pathogenesis, the more familiar way of thinking about it in terms of disease process. We do have additional objectives, which is really to really make this as open as possible, so that we are hoping to share the data set and associated tools and standards and guidelines to really make ourselves as the flagship data set, and then really provide blueprints in terms of how to generate a data set like this. And we want to train the next generation of the workforce that Dr. Baxter is going to highlight later. And we want to advance understanding of team science. So why do we focus on type 2 diabetes? It's because more than 37 million Americans have actually diabetes, and that is about 1 in 10 people, which is a remarkable number. And more than 1 million per year actually die because of diabetes alone, and it's the 9th leading cause of mortality worldwide. And we really wanted to focus on type 2 diabetes because it is such a systemic disease that involves eyes, brain, heart, and a lot of multi-organ diseases. And we thought that by working on this, by developing a framework on collecting data to study a very multifactorial disease, we thought that this would be really helpful for our field. And the concept of how are we collecting this data? And then Jack mentioned that we are going to collect a multimodal data set from 4,000 people. And then I just mentioned, well, I want to study salutogenesis. The concept really is based on the single-cell RNA-seq technique that to analyze the single-cell RNA technique, there have been very successful machine learning progress to analyze a single-cell RNA technique called pseudotime manifolds. So the pseudotime manifolds techniques is that when you have all these different types of data sets, and then you are making all the cells that are similar, very close to each other, that are very different, very far apart from each other. So even though you're developing, you're performing single-cell RNA-seq from one sample, you can kind of really spread out the differences and the similarities between the samples, and then create the pseudotime manifold map. And essentially, you're able to visualize what happened during the embryonic stage, and then how did they become differentiated into, you can think about a healthy stage versus disease stage. So this is the idea behind us creating this data set to study pathogenesis and salutogenesis. So I'm going to quickly go over this since we already mentioned this. The 4,000 participants are coming from University of Washington, UCSD, San Diego, and University of Alabama at Birmingham. And we are hoping to balance all the data set as much as we can because we recognize that really big limitation of currently available data set to train machine learning is that all our data sets are highly imbalanced. So we'll equally balance into men and women, a biological sex, and then Asian, Black, Hispanic, and white. And then also we are dividing equally among the diabetic disease stage as 25% non-diabetic, 25% diet-controlled diabetic, 25% medication-controlled, and then last quarter, insulin-controlled diabetic. And we are collecting data from heads to toe, literally heads to toe, because we are starting with cognition with MoCA. And then we are a little bit eye heavy in terms of a lot of retinal imaging, beautiful retinal imaging that allow us for us to really obtain a sophisticated data. And then 12 lead EKG, and then blood work, and then we'll mention more of a exciting data set such as continued glucose monitoring, fitness tracking, environmental sensor data. And then we are also banking samples so that people can request samples such as Buffy coat, plasma serum, and then PBMCs. And we are planning on doing whole genome sequencing at the end of 4-year period as well. So this is an extensive protocol, but I wanted to kind of show how extensive it is, and then extensive the data that you could leverage for your research questions. I want to highlight some of the questions as CESD on depression, social determinants of health. We are using FANX, visual impairment, and also the environmental sensor data that we'll mention later on. So how are we doing? So we collected our pilot data until November for about 3 months last year. And we have actually released our data set from pilot. It's from 200 participants, 204 participants. And I just wanted to kind of show how big it is. Just from 204 participants, we have over 500,000 of glucose measurements, over 1.2 heart rate measurements, 33 million air pollution and light spectrometer reading, and then lots of data. And then we already banked over 2,500 tubes of biospecimens from this pilot data. And this pilot data alone is about 311 gigabytes, and then the full data set of 4,000 people should be about 8 times, should be 20 times more. So now the question is, wow, that is a lot. And then I mentioned that this is a pilot study, and then now we are getting ready for us to do the year 2 release data, which will have over 1,000 people's data. And so then what are some ethical considerations? One, it's return of results instead of finding, advisory council involvement in terms of us getting the advice, continuous feedback from how we are doing. And then how are we planning on engaging with past and future participants and data access and sharing? So let's go over that. So at the end of each visit, we share this incidental the return of results card. And this was really based on what all of us does in terms of the measurements that we can get. And then we let them know what were their blood pressure, what were the visual acuity. And we kind of let them know, if your vision is less than this, we suggest that you see an ophthalmologist, etc. So then we are giving their information right then. And we obtain 10-day worth of continuous glucose monitoring combined with 10 days worth of fitness tracking with Garmin watches, and then also 10-day worth of environmental sensor. And after they return those equipments, we send them the results of their continuous glucose monitoring. And we send them how to analyze or how to understand this guidelines in terms of this is a glucose management indicator, which is essentially kind of estimated of hemoglobin A1C over those 10 days. And we provide this type of information so that participants can know what our reports mean, but then also can take that to their primary care doctor if they want to discuss this further. And this has been a really good, well received by participants, because participants are really interested in knowing what their level is like, especially for people who are actually not diabetic, and then they don't have a good way of getting continuous glucose monitoring. And then so this has been a huge help and really attractive for the participants. One of the things that I wanted to mention before I move on to the advisory council is that we do check for incidental findings. So things that we weren't worried about, we are not really checking, but if there are any signs of immediate vision or life-threatening conditions that we find, then we would immediately get in touch with the participants. So for example, the things that we would look for will be tumor in the eye, or retinal detachment in the eye, or optic nerve edema, swelling in the optic nerve. That could be, related to maybe potentially a brain tumor, or lots of, pressure in the brain. So then one occasion, we actually had a patient that had no symptom, and the participant came in and then was getting photos taken, and then we picked up a retinal detachment that was vision threatening. So we called that participant immediately, and then he went to on the way home, he went to the emergency room, and then we actually ended up doing surgery that night. So things like that, we are referring participants with incidental findings if we find them. And we got a lot of advice from bioethicists and then our team in terms of how to set up our study and then also communicate, and then how to do the consent. And we have an advisory council, which is we tried really hard to make all of them really representative of all the participants that we are trying to recruit. So making sure that Asian Americans, Hispanics, and Black are also represented in the council, and then women are represented in the council. And we picked up members from each collection site, and then everybody went through an interview, and then we make sure that they are compensated for their time, and they are really our advisory committee or council in terms of questions and if we needed any feedback from us from them to make sure that what we are thinking is correct, or what we are thinking is in line with what the community members and potential participants would think as well. So this is an example of our feedback. So this is actually the initial recruitment letter that we sent to our participants, and this was like the initial interface that potential participants were receiving. And we asked our advisory council, what do you think about this? Could you, give us any feedback? How could we improve? And after their feedback, this is the improvement that we made. And then I think this, really highlights the big difference that they are making in terms of our study. And then they are really helping us to design how to get more values to the participants. So I mentioned the engagement with past and future participants. So the advisory council is actually really helping us to try to come up with a way. So right now, we obviously have our website. And then also we are asking all the researchers that are requesting our data to share their research questions. And then so our participants can actually see what their data is being used for research. And then we want to do updates on our website, any new publication, any results that we find. And with the advisory council under the leadership of Sam Hurst, we are trying to plan a virtual seminar where everybody is invited. If you're future or past participants, and then we can kind of go over our progress and then any of the results so far. So that is one of the major milestones for year 3. So we also mentioned that, the last item, really big item is, how do we make this data accessible and then, share as much as we can while making this, while ensuring the safety and then protecting the privacy of our participants. So I'm going to move that into Bhavesh's next set of presentation. But before we start, Bhavesh is going to go over. Well, before we discuss the access and sharing issues, we need to first make sure that data is actually prepared for analysis and AI ready. So Bhavesh.
Yeah, thanks, Cecilia. I actually flipped it around. I'm going to tell everyone first how to access the data set and then tell them what it looks like. Yeah.
Bhavesh, do you want to share your screen? Okay.
Yeah, I'm trying to do that right now. Can you all see my screen? Yeah, okay, great. Yeah. Hey everyone, I'm Bhavesh, and I'm, like Cecilia mentioned, there was a lot of consideration that went into how we share the data. So I'm going to talk about that next. But first, I'll tell you how to access the data, since I'm sure you're now all excited about it. And so the AI-READI data set is shared through a new platform we are developing as part of this project called FairHub. And I'm going to talk about that platform soon more. But to get to the datasets page on FairHub, there's different ways you could do that. To our project website on AI-READI.org, to our dataset documentation website at docs.ai-ready.org, or even directly through fairhub.io. And irrespective of the route you select, you'll end up on this page, which is our datasets landing page on FairHub. And there you'll find different tabs that provides various information about the dataset. So you can learn more about the dataset itself, the content, the study, the provenance of the data, how it is structured, and so on. And once you're like, hey, yeah, this is a dataset I'd like to use, you can click on this access this dataset button. That will take you to this page where you have to go through actually several steps before you eventually get a link to download the dataset. And so you may wonder why there's so many steps to get to the dataset, and especially since our public access data doesn't contain any PHI. So based on our consent, all the variables that are in the publicly accessible data don't contain any personal health information, but we also excluded some additional variables such as race, ethnicity, and sex. So theoretically, we could share it in a repository where you can just click on the download button and download the data. But we took the opportunity in this project to kind of rethink the open data sharing approach, and we came up with this method we call the Swiss cheese model of open data sharing, or as a vegan, I like to call it the vegan Swiss cheese model. And the idea here is that we introduce several layers between you and getting access to the data, with each layer intended to prevent any risk to the participants' privacy, but with understanding that none of these layers are foolproof, so each of them might have some holes, but then in series, they might be more and more effective at preventing any risk. And one of our reasons for creating FairHub was that we couldn't find any repository that supports such an access process. And so this is why one reason why we developed FairHub. And so when you try to access the data set on FairHub, the first thing we do is ask you to log in through an identity verified system. And so this is a way of logging where we can get your real name. And so, for instance, currently we integrate with CI Logon, which allows you to log in through your institutional email, so such as a .edu email. One limitation of that is that it doesn't support access for non-academic users. So we're also in the process of integrating with the NIH's Researchers Auth Service, or RAS, which would allow even non-academic user to access the data, for instance, through the login.gov ID. Once you have logged in, the second step is for you to read and agree to our license agreement. And here we have developed a new license that explicitly prohibits re-identification of study participant or harm, but also prohibits distribution of model that may copy or memorize the data, and prohibit distribution of the data. So anyone wanting to get to the data has to go through these steps to get to it. But the license does allow commercial use. And we're really hoping that this license may eventually become kind of a standard when sharing clinical research data that's expected to be used by AI and ML models. After the license, the next step is for you to type out character by character several attestation, where you test different things such as not attempt to re-identify participant, distribute the data, and so on as per the license agreement, but also agree to use the data only for type 2 diabetes-related research, because our consent form says that the open access data will only be used for type 2 diabetes research. And the idea here is to kind of create a social contract that target directly the individual user who's trying to access the data, when the license is often seen as something that target more the institutions. And if all of that is still not enough to kind of warn you about not doing anything wrong with the data. The last step is watermarking. So as part of the attestation, you also attest that your data would be individually watermarked. And so what we do here is each user gets a uniquely watermarked data set tied to their identity that we get from the first step where you log in. And so if the data set appears somewhere online, we can track down who leaked it. And of course, we do kind of recognize that even with all of those layers, there might still be risk. And we do mention that in our consent form. But the idea here is as we learn more throughout the project, we can always add more layers to this process. And as a French, I'm always up for more cheese slices. I'd be happy to do that. And so now that I explained to you how to access the data set, I'll talk a little bit about what do you get when you download this data set, right? What does it look like? Cecilia mentioned there's more than 20,000 files. So when you download it, is it a pool of files? Does it have more structure? What format the data comes in, and so on? And all of those considerations were made keeping one main thing in mind is to make the data AI ready. And so we ask ourselves, hey, what does it take for the data to be ready for AI and ML model? And we thought that at the high level, it takes two main things. First, the data has to be ethically sourced and shared. And this is what Cecilia and I already covered. And the other side is to have the data technically ready for AI and ML model, meaning that it should be in a file format that's machine friendly, have metadata in a machine friendly format, and so on. And all of that is covered by what's called the FAIR principles, where FAIR stands for findable, accessible, interoperable, and reusable. These are a set of principles that were published in 2016. They were the results of a large-scale effort by researchers, funders, scientific publishers in all fields of research and all over the world, who came up with 15 principles that kind of guides how you can make your data optimally reusable by not only humans, but also machines. And I won't go through those 15 principles, but typically kind of implementing this principle require 5 main items. First, structure the data according to a consistent folder structure, so it's easy for humans and machine to navigate through the data. Secondly, use standard file format, so use file format that anyone can ideally easily open and use. Include metadata using standard format and vocabulary, so you can describe your data in a way that anyone can understand it. And provide the data reuse license, which I already mentioned. Without a data license, the data is not reusable. And finally, share the data on a suitable data repository, which would make the data work toward making the data findable and accessible. And we try to cover all those 5 elements in our data set. And so when you download our data set, this is what it should look like. There's one folder per data type and a bunch of metadata file. And this is based on a standard we developed, and we are developing actually. It's still work in progress as part of this project that we call the clinical dataset structure, which is a standard that intended to consistently organize clinical research data and metadata, so that ideally, if everyone is following it, all the data sets should be looking something like that, have the same naming convention and so on. So it would make it easier to merge 2 data from different studies together. And it doesn't just specify a structure at the high level. So but it also provide a sub-level structure. So if you open a data type folder in our data set, there'll be 1 folder per modality, then 1 folder per device, and then 1 folder per participant. And when you open that participant folder, it should have a data file in a standard format related to that data type. And so, for instance, for clinical data, we followed OMOP. For retinal imaging, we followed DICOM, and so on. And so we here use a data format that anyone should be able to access through openly available tools. And as I mentioned, there's also a set of metadata file that provides information about the data set, the study, the structure of the data, and so on in both human and machine-friendly format. And so, for instance, one of those metadata files is a participants.tsv file that for each participant tells you what study group they belong to, the age, the study visit. We also provide our recommended split. So if a data from a participant should be used for training, testing, or validating the model. So you have a balanced set for each of those elements when you are developing your model, and also provide which data type are available for each of the participants. And so there are many other such metadata files that are included that hopefully once you download the data set, you can fully understand what it is about, how to use it, and get basic information through those metadata files that would help you use it and kind of guide you in using it for your purpose. And so the second reason we developed FairHub is to help us into structuring the data in this way. As you can imagine, if we were to do that manually, it would be very time-consuming and difficult to do. So we are developing a FairHub with two purposes. And I already talked that one element of FairHub is the data access portal, where you can access the data through this novel kind of method that we have implemented. But there's also a data management site where researchers who are collecting the data can upload their data as they are collecting it. And FairHub includes a bunch of tools that helps format the data as automatically as possible into the structure I just described, create those metadata files and so on. And then when it's ready for a release, they can also easily push it onto the data access portal while obtaining a DOI, having this metadata that index it into Google and Google dataset and so on automatically. So it makes the data set fair. And I believe now I'll pass on to Sally, who's going to tell you more about other resources and effort that we are doing around this data set. Thank you.
Thanks, Bhavesh. I'm just going to be sharing now as well here. All right. Go to slideshow mode here. All right, so now that you've had a chance to, learn about kind of the setup and the premise and content of the data set, I'm going to talk a little bit about what documentation is available to help you with actually working with the data set once you get access. So this is all available at our website called docs.ai-readi.org. And you can see there's two portions, and Bhavesh talked about the portion of accessing the data set already. And what I'll be talking about is the documentation portion. So as you go in, there's a general area to learn about the documentation and what it entails. We do provide information about O2, the terms of the data usage license and how to cite data sets. This is a very brief overview, and I would encourage everyone who's using the data set to really look at the full license terms as well, which we have hyperlinked here also. This is an important part of initially getting access. As Bhavesh mentioned, there's a series of attestation statements that you have to put in, and part of it is around what prior training you have. We want to make sure that people who are using the data set, don't use it for nefarious purposes and are sort of honoring all of the typical human subject research protections that we would expect for anyone using data that has originated from human participants. We have training resources on basic research concepts as well as human research subject protection. If you've had this through your institution already, you certainly don't need to do it again here, but you do have to attest to having completed those in the past. If you are now coming from an academic institution or you haven't had to do those trainings before, we have a slew of different resources to help you complete that training. And we're also adding a section specific to AI ML with the upcoming data release. We do have a health sheet as well. So there are a couple of different frameworks for documenting data. You might have heard of things like data sheets, health sheets, data nutrition cards. And we ended up using the health sheet because we thought that this was most in line with the type of data that we have in this study. so it has a standardized framework around the summary of the data set, the rationale, how many participants, safeguards are in place to protect considerations of privacy and confidentiality and so forth. What kind of data quality control measures were taken. So we followed that framework, and we have pretty detailed information across all of those domains for this data set. So that can also serve as a starting point or a reference for you as you get oriented to this data. And then if you look on the left here, we have various data domains and a whole range of documentation specific to each data domain. So depending on what you're interested in studying, you might focus in on some aspects more than others. For example, in the clinical data, we have a whole area of documentation explaining the various laboratory values that are included in the data set. We have explanations of all the questionnaires and surveys that our participants have completed, including the individual items, so that you can see what kind of data elements are there and available for analysis. We have information on each of the devices in the protocol, the retinal imaging modalities that Cecilia mentioned earlier when talking about the protocol itself and the data collection. And so for each of these, when you click in, it'll give you an overview of the background of that data domain, what the clinical rationale is, how it's used in clinical practice and in research. We do talk about data processing, like when you get a copy of the data set, how are the files formatted? What is the data structure? What's kind of the file directory or pathway to get to the data that you need? We talk about standardized terminologies for the data, any metadata that's included in example outputs. So for example, at the end of each data documentation page, you'll see a list of like the column headers and what they mean, as well as example values. So this is what I was talking about there with the metadata, right? Is sometimes you get these CSV files and it's all like it seems like Greek, right? Like you can't necessarily decipher it without a guide. And then that's what we were hoping to provide here, was a guide of, okay, here are the variable names. Here's what each variable describes and what are the example outputs and what they look like. The goal here is, hopefully, as you're new to the data set, you have this orientation or guide to help you get started and get oriented and make it easier to do the actual analysis. So just as a status update of how many people have accessed the data set so far, we released the pilot data set in October. I believe that's or as of October 1st, 2024, we released it in May, my apologies. Now, as of October 1st, 112 research organizations worldwide have downloaded this data set and started exploring it and using it. We have another sort of batch of data for the participants that we've enrolled since the pilot that will be anticipated for release in November of this year, so just next month. And this will include data from over 1,000 participants. So there'll be a big increase in the data set size and a lot more data to use for various research and AI applications, hopefully. We are going to be working, looking ahead at data use agreements and kind of what the framework will look like for sharing the restricted data set. So as you might recall, Bhavesh mentioned that there were some of the variables that have not been included in sort of the public-facing data set for reasons of preserving patient or participant privacy and so forth. We are working on ways to figure out how to share those restricted variables down the line with additional safeguards. And so just pivoting a little bit, the documentation resources and training resources that we have there are available to basically anyone who's interested in learning about the data set. And certainly those who are getting access to this data set will hopefully find those helpful as they start analyzing the data. But I also wanted to highlight some more sort of intensive training opportunities for people who might be interested in, doing more than just working with the data, but be interested in getting some skills development and some mentorship in AI/ML and biomedical research. So as part of the Bridge to AI initiative, there has been this emphasis on training and workforce development and building our future workforce in AI, and making sure that we have opportunities, especially for trainees who might come from populations that have been traditionally underrepresented in this field. Our grand challenge of the AI-READI group decided to endeavor on a full-time mentored research program. We are now in our second cohort, and we are recruiting for our third cohort, which will be running next year from August 2025 to July 2026. We do require at least an undergraduate degree, so at this time we're not taking high school students and college students, but anyone undergraduate degree and above is eligible. One unique feature is that you do not need to be a U.S. citizen or permanent resident to apply. That is a restriction for a lot of NIH training grants, and it is not one for this particular program. There is a full-time stipend support at standard NIH levels, and it includes an in-person 2-week summer programming boot camp, by the beach at UC San Diego. And then our trainees are placed with AI-READI mentors across the country throughout the year to work on projects, be involved with various initiatives across our Grand Challenge, as well as additional mentored research projects, at the discretion of their mentor. Our applications are now open, so please spread the word to any trainees who might think are interested in this. The other opportunity that's sort of just kind of fresh off the press is an emerging collaboration we have with the AIM Ahead Consortium. And this is another large NIH consortium that's aimed at promoting diversity in AI and ML. And this one's a little bit different than the full-time internship in that it's more of a part-time opportunity, about 5 to 8 hours a week. There are hands-on workshops to, basically have support and kind of hands-on guidance in getting through the access to the data set and working with the data set and developing a project. It'll run for 9 months. We are still finalizing the call for applications, but we anticipate that they will open either later this month or early next month, and it'll be for January 2025 start. There is some stipend support, so about $8,000, as well as travel and publication support. For this one, you do need to be a U.S. citizen or permanent resident to participate, and we look forward to being able to share more information about this soon once it's finalized. If I were just to wrap up before we open up for discussion and questions, we're thrilled to share that the marker paper for the overall project and Grand Challenges have been approved for publication at Nature Metabolism, and we have several additional manuscripts in progress about various aspects of the project, including team science components, data standards, some of the devices, including a novel environmental sensor that is part of the protocol. Some of the things that Bhavesh mentioned about fair data principles, how do we have data set documentation and transparency, open data, and then as well as some manuscripts around our training program, curricular development, and our boot camp experience. So, a lot of exciting things to come. We're excited to disseminate our experience with this project. And looking ahead, we want to ensure broad accessibility, to the scientific community, continue, as I mentioned, dissemination and outreach efforts, not just through publications, but through webinars and events like this. We're also partnering with other large consortia like DKNet and AIM Ahead to really spread the word about this new data resource that will hopefully be useful to a large group of folks who are interested in doing work in diabetes and health research in general. And we really hope that this will promote our understanding of salutogenesis in diabetes. So just in summary, AI-READI is a multi-center, multi-dimensional data set, so many different data types, so many possibilities, and uniquely positioned, I think, to answer a lot of interesting questions, given the diversity of the cohort, and really great representation across racial and ethnic groups, sex, and diabetes severity. We're hoping that people will be able to use this data set for a wide array of possible research questions. And a large part of our emphasis has been on fair data principles and trying to facilitate access to the broader community. I think with that, we're wrapping up the structured talk portion and have some time left for questions and answers.
Thank you so much, all of you. At this time, we will open this session to the audience, and please ask away.
I saw that there are some questions in the Q&A. If in the future you discover that the data from FairHub could inadvertently include PII, then can you elaborate on what steps are to be taken to mitigate the resulting data compromise? Cecilia, I saw you were typing. Do you want to just answer it live, maybe?
Yeah, I mean, I was saying that we would pull it immediately from the public repository. So that would be what we would do. Let me just do that. And then we do a lot of vetting and checking before we do our annual release to make sure that there's no PIIs. Bhavesh, do you have anything to mention from like the tools perspective, or yeah?
Yeah, I think because we do identity kind of verified login, we could reach out to all the people who had access to the data and request to kind of destroy the data and kind of download the new version that we will release after addressing the issue. I think, and then in any case, our license do prevent from even if anything is linked to using that to re-identify any participant.
All right, I saw another question in the Q&A. What is your future strategy to improve geographical coverage and diversity? It seems that the people living in the Midwest are not represented in this data set. So I can take that one. I think, we would love to all have studies that have nationwide coverage, sort of like, All of Us program has enrollment sites across, I think, most states in the country. But, for us, it was kind of not within scope, and with the funding and budgetary constraints that we had in place, we could only fund, 3 data collection sites. And so at this point in time, we don't have plans to add another clinical site. I think the offshoot, the balance between, we don't have necessarily a huge number of participants across all geographic coverage. But the flip side of that is, for the participants we have very dense, granular, well phenotyped data. And so I think that there are, benefits and challenges to those situations, where you have a large amount of coverage and a large amount of people, but, mostly diagnosis codes or things like that, and maybe not so much granular device data, imaging, all these things that are very time intensive and difficult to obtain, versus a data set like ours, where we do admittedly only have a limited number of clinical sites, but we have very in-depth, very detailed information about those individuals. So depending on your research question, each of those data sets might offer relative advantages and disadvantages. But in any case, that was kind of what we saw as something missing from existing data sets, was this super granular, dense, detailed phenotyping, and that's what we hoped to achieve with our data. It looks like there's a question about, are dates shifted in the de-identified data set? Cecilia?
Yeah, I can take that. So they are not shifted because we don't have any clinic visit dates. All the dates that we're sharing is research visit date. And then the IRB, and then the prime institution, the main IRB is the University of Washington, and then we do not deem the research visit date as PHIs.
Perfect. I think that relates to Grace's question about give an example of the resolution in which the data will be associated with specific conditions in which the data were collected. Will it be easy to synchronize, for example, if a weather or unexpected event occurred during the time of the collection? I think a very timely question given the recent, sort of catastrophic weather events, and we want to, acknowledge anyone who's in the affected regions. We certainly, our thoughts are with you. But yeah, I would, following what Cecilia said, because they're real dates. For example, with the environmental sensor, we do have data collection where the patients take that, or participants take that home with them for 10 days. Those will reflect, real time. So I think the synchronous nature of the date time information is an advantage for the type of question that you're posing, Grace, because I know with other data sets that are shifted, like all of us, that's sometimes a challenge because they date shift, randomly between 1 and 364 days. And so you can't necessarily synchronize that or line it up with major changes in health policy, environmental events and so forth. But I think for our data set, you can, because they're sort of real date timestamps.
Yeah, so one of the very nice, research question would be like, what would be the temporal fluctuations? And then also, like Dr. Ping mentioned, also looking at the environmental data of the community of that geography versus the home environmental pollution or temperature, all those data. I don't think there are any studies that have really done home environmental data specifically. So I think that would be a very nice, interesting research questions that could be answered with this data set.
I don't see any other open questions in the Q&A. We do have 3 minutes, so we could probably take maybe one or two more. Okay, so from Henning, I've seen a very well set up, thoroughly documented study with which without a doubt produces data suitable for AI-based analysis. Are there any aspects specifically to support use in AI, for example, in the files, like pre-calculated embeddings? Bhavesh, do you want to take this one? I know you mentioned some of the training and validation sets and so forth. That might be worth.
Yeah, I'm not exactly sure what it means, but I wonder if it's asking about maybe file level metadata or information. And we do have those, for instance, in the DICOM file, there's the headers that include certain information. The environmental sensor file also do the same. So between the raw data that's collected, we do have, and we now have it integrated in FairHub, data processing pipelines that do all the transformation and that transform the data into a format that we deem would be suitable, at least so far, for AI use. And that includes adding additional metadata. Actually, for some of those, we also link to the REDCap data that we pull from there and add it to the files to provide additional context and so on. So I'm hoping that's what the question was about.
And then also, we provide the suggested split of the data set with each release in terms of, the training and validation and test set to ensure that, major demographic or the, data sets are balanced. So we're hoping that will be really useful.
All right. Jack and Pepe, I know we're coming up on time. So
yeah.
Guys.
Thank you. Thank you, Sally. Thank you, Cecilia. Thank you, Bhavesh. This has been fantastic, very educational, very insightful lecture. We appreciate amazing contributions and your launching this series of 4 lectures that's coming up. And you've really set the bar very high. And we thank you again for everything you 3 have contributed today. And so I'm handing it to Jack, who's going to remind.
Yes, I just wanted to share real quickly. We invite all of you to join us for the upcoming sessions on November 7th, 14th, and 21st at 12 p.m. PDT, where we will explore the remaining Bridge2AI Grand Challenges, introducing you to their data sets, and that's from Voice, Chorus, and CM4AI, respectively. Thank you all for dropping in, and we really appreciate you, the lecturers today, for preparing this for us.
Thank you so much for having us.
Thanks.
Thank you
Bye.
so much. Bye-bye.